So, good morning to all. Good morning, everybody. So, hope you are all doing well. So, is my voice audible to you? Yeah. So, good morning to everyone. So, today's class is our almost last class for this online. Because I think CBSE students, they have their 10th class examination, like one more paper they have to write. I think maths exam they are conducting again, right? So that's why the management has decided to discontinue these classes for a while, and you may be asked to come for offline classes after 15th of May, mostly after 15th of May, okay? So till that, the state board students can take rest, because you are in the classrooms, like you are continuously, following our classes as soon as your 10th class examinations is completed. So we know that you are already feeling a lot of pressure. So that's why, so the management has taken a decision of giving you, some kind of rest before you come for offline classes. So once you come for offline classes, then you have to you have to work, at most highest energy levels. So most probably today may be the last class, and from Monday onwards you may have, a short vacation, and you may be asked to come for offline classes after 15th. Yeah, thank you. So this is about your class and holidays. Now let us go into the details of the cell unit of life. In the last class I discussed about bacterial structure, right? So structure of bacteria, prokaryotic cell. So today, after, you can ask all your doubts at the end of our class. Like I'm asking the technician to stop messaging. So your chat box is already closed. Now you can ask all your doubts at the end of our class, like last 10 minutes. Now listen to me carefully. So I told you in the last class that this bacteria, this is the bacterial structure I gave in the last class, right? So in this is cell wall, then there is a glycocalyx capsule, that is glycocalyx. Then you can also see pili here, fimbriae, and this is flagellum. So these are all the things we already discussed in the last class, right? Now let us go into some other details. Now these are the ribosomes. In the last class itself I told you that there are 70 ribosomes in the bacterial cell. Next, when it comes to the eukaryotic cells, as you know, eukaryotic cells have a well-defined nucleus. So now you can see this is the nucleus with nuclear envelope. In all the cells, like it may be protistian or fungi or plant or animal, in all of them there is a nucleus. Now this is the plant cell structure with cell wall, nucleus and various types of organelles. And you already know that only eukaryotes have membrane bounded organelles. Membrane bounded organelles are present only in case of eukaryotic cells. So whereas in prokaryotes like bacteria, they have only one organelle, which is amembranous organelle, that is ribosome. Whereas these animal cells don't have cell wall. You can check here, there is no cell wall. their cell is directly surrounded by the plasma membrane. Of course, it is a animal cell like human cell of intestinal cell. That's why you can also find microvilli here. Okay. So next, now see this diagram. This is the structure of plasma membrane. I told you in the last class that bacterial cell as well as eukaryotic cells contain plasma membrane, which is structurally similar. The same kind of structure you can find. So if you take this cell wall, so in the plasma membrane, so in the plasma membrane, this is lipid part. You can check here, this is lipid bilayer. So in the plasma membrane, lipid is present in 2 layers and proteins are also present. Arrangement of lipids is considered to be symmetric and arrangement of proteins is considered to be asymmetric. There are a lot of details are there about this plasma membrane. I am not touching all those details, just I am giving you basic details. So basically this plasma membrane is composed of lipids and proteins. Apart from lipids and proteins, there is carbohydrates, that is sugars. You can also find cholesterol. You can check here, there is cholesterol. Cholesterol is also present in the plasma membrane. Understood? Even the composition of this, sugars as well as lipids as well as proteins is not same for all plasma membranes. It varies from species to species. If you take the plasma membrane of animal cells like erythrocyte or red RBC of animal cells like human, it contains around 52% of protein and 40% of lipid. How much? 52% of protein and 40% of lipid. So this is the composition of protein and lipid. And proteins are again different types. There are some proteins that are present. See, you can check this diagram, it is more clearer. So if you look here, we also again have different types of proteins. Look at here, this protein is embedded in the membrane. Embedded means it is completely inserted in the membrane. Now this kind of proteins are called integral proteins. What do you call them? Integral protein. Understood? This integral protein, again 2 types. Look at here, this protein is extending from one side of the membrane to other side. This is wholly embedded or totally embedded. Now look at this protein, is it completely embedded? No. So only a portion of the lipid, into a portion of lipid the protein is embedded, remaining portion is outside. Okay, this is called partially embedded. Partially embedded, wholly embedded. There is also some protein like peripheral proteins. See, you can check here, this is a peripheral protein. So we can have peripheral proteins, integral proteins again different kinds like totally embedded, partially embedded. Now different models have been proposed to explain structure of plasma membrane. The most accepted model is fluid mosaic model. What is it? Fluid mosaic model, and it was proposed by Singer and Nicholson. Who proposed it? Singer and Nicholson. They have proposed this fluid mosaic model. According to this fluid mosaic model, in the plasma membrane, lipid exists in quasi-fluid form. Which form? Quasi-fluid. QUASI. Quasi-fluid in the sense semi-fluid. Semi-fluid form, in which proteins are floating. Just like, simply I can give you one example. Icebergs floating in the ocean. You can take as example for this plasma membrane structural details. Icebergs floating in a ocean. Ocean is composed of water, right? This water you can compare with lipids. In that lipid ocean, now icebergs are floating. Similarly, in the plasma membrane too, there is lipid ocean in which proteins are embedded. And do they have any specific arrangement, these proteins? No. They have irregularly arranged. Look at here. They are not staying at one particular region. They are spreading all over the lipid. That's why I said arrangement of proteins in the plasma membrane is asymmetric. There is no symmetry in that. Whereas lipids have symmetrical arrangement. Understood? You can also take another comparison. Payasam, which is prepared at your home by your mom. It's payasam. It's a semi-fluid liquid, right? In that you will be putting cashew nut. You can take this cashew nut as an example for protein. That semi-fluid liquid, whatever it is made, the payasam, you can compare with lipid. So these cashew nuts will be floating in semi-fluid liquid of this payasam. Similarly, this plasma membrane is also having a semi-fluid, what kind of layer? Lipid, in which the proteins are floating. As this lipid is quasi-fluid in nature, therefore proteins can show lateral movement. Look at here, this protein can move this way or this way, and proteins can show rotational movement. Rotational movement is also possible. Say for example, if this lipid is solid, is it possible for protein to migrate from one region to the other region? No. Isn't it? As lipid is semi-solid or semi-fluid, hence proteins can exhibit lateral movement. So this lateral movement of protein is an indication to say that this plasma membrane, lipid part of the plasma membrane is having quasi-fluid nature. And this quasi-fluid nature of plasma membrane is also responsible for various kinds of functions. Look at here, this fluid nature of the plasma membrane can help in cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, all such kind of functions are carried because of this quasi-fluid nature. Understood? So this is what you can see. Of course, plasma membrane is having several other functions, like it helps in transportation of materials from one side to the other side, and plasma membrane involves in active transport, passive transport. Plasma membrane is selectively permeable and it helps in simple diffusion, osmosis, facilitated diffusion. These are all the functions carried by the plasma membrane. Similarly, let us take the cell wall. See, just I am covering all the topics, so I am going little bit faster because we have to complete this topic today. Just I am giving you basic details. I am not going in depth of every topic. See, plasma membrane itself need 2 hours to complete. In our offline classes, we are going to discuss about this plasma membrane for about 2 hours. So just I am giving the basic details. I am not touching any inner depth, in-depth details of these topics. Now come back. Cell wall. So when it comes to the cell wall, is it living or non-living? Non-living. A rigid structure present on the outer surface of the plant cell. You can check here. This is cell wall present on the outer surface of the plant cell. And this cell wall, in case of algae, the cell wall is composed of cellulose, galactons, mannans and minerals like calcium carbonate. Whereas plant cells contain cellulose, hemicellulose, pectin and protein. In case of plant cells, the first formed layer of cell wall is middle lamellum. What is it? Middle lamellum. Look at here, look at carefully. So middle lamellum is the cementing substance present between two adjacent cells. Now this is middle lamellum. Can you see this purple colored line that I have drawn between the two cells? This is one cell, this is another cell. Between these two cells there is a middle lamellum. It is a cementing substance that can hold adjacent cells together. Okay, that means simply we can say it acts like a glue for the cells to attach with one another, or simply to say it is intercellular ground substance. And this middle lamellum is actually responsible for formation of tissues. Okay, if middle lamellum is absent, then cells cannot attach with one another. They get separate and they remain as a unicellular structure. So you cannot see this middle lamellum in unicellular organisms and also in colonial forms. Then where do you see them? Only in multicellular organisms. Understood? And what is its function? It acts like a glue, or it holds the tissues, or holds the cells with one another. So that is the importance of this middle lamellum. Okay. So it is composed of calcium pectate. Of course, magnesium pectate is also present. Then on either side of the middle lamellum there is primary wall. So primary wall is present. Now you can see this layer, this primary wall. So this primary wall growth diminishes as the cell matures. The secondary wall is formed inner to this primary wall. Inside of this primary wall, the secondary wall formation takes place here. Now think that this is primary. Now the secondary wall formation takes place here, inside. And this secondary wall is rigid and it do not allow the cell to expand. But primary wall can allow the cell to expand. So this primary wall, secondary wall, middle lamellum, all these three layers you can see in a matured plant cell. In a meristematic cell, you can see only middle lamellum as well as primary wall. So this is what you can see with regard to the cell wall. Next, cells also have endomembrane system. Endomembrane system in the sense, there are some organelles in the plant cells whose functions are coordinated. That means their functions are interconnected. What are they? That includes endoplasmic reticulum, Golgi complex, lysosomes, vacuoles. This four comes under endomembrane system. What is it? Endomembrane system. Whereas the organelles like mitochondria, chloroplast, as well as peroxisomes, they do not come under endomembrane system. Why? Because their functions are not coordinated with the functions of organelles present in endomembrane system. Understood? And one more interesting thing is, listen to me carefully, this endoplasmic reticulum is the precursor for the production of Golgi complex, or simply to say, Golgi complex originates from endoplasmic reticulum and lysosomes originates from Golgi complex. Lysosomes originates from Golgi complex. Endoplasmic reticulum produces Golgi complex. Golgi complex produces lysosomes. Vacuoles can be formed Golgi complex as well as endoplasmic reticulum. So their origin is also interconnected. Got it? Their functions are coordinating. Their origin is also interconnected. That is the importance of this organelles of endomembrane system. Now let us see brief understanding about these organelles. Now if you take the endomembrane system, endoplasmic reticulum, look at here. Endoplasmic reticulum, it originates from outer nuclear membrane. So this is the outer nuclear membrane. It originates from outer nuclear membrane and extends up to the plasma membrane. So it is simply to say a network of tiny tubules. Tiny means small tubules, which is scattered all over the cytoplasm, and it divides the total cytoplasm into two distinct compartments, like intraluminal, like inside the endoplasmic reticulum, that is intraluminal, extraluminal, that is outside the endoplasmic reticulum. Understood? There are two compartments are there in the cells, and apart from that, this endoplasmic reticulum is again two kinds, on the basis of whether their surface is studded means attached with ribosomes or not. If the surface of endoplasmic reticulum is studded with 80S ribosomes, which ribosomes? 80S ribosomes, then it is called rough endoplasmic reticulum, that is also called RER, or granular endoplasmic reticulum. If there is no 80S ribosomes on the surface, then it is called smooth endoplasmic reticulum. So this rough and smooth endoplasmic reticulum also have different functions. Rough endoplasmic reticulum, as their surface is studded with ribosomes, it can help in protein synthesis. Smooth endoplasmic reticulum, it helps in lipid synthesis. It is the major site for lipid synthesis, and in animal cells it also helps in synthesis or synthesizing lipid-like steroidal hormones like testosterone, estrogen. Such kind of hormones can also be synthesized. So that is the importance of RER and SER. Next, when it comes to Golgi complex, I already told that Golgi complex originates from endoplasmic reticulum. Remember, the Golgi complex as well as endoplasmic reticulum, their structural components are almost same. Both have cisternae. Can you see this structure? This is called cisternae. They also have tubules. They also have vesicles. And of course, additionally, the Golgi complex contain vacuoles. Golgi vacuoles are also present. Okay. Cisternae is a long, flattened, disc-like structures. So these are all disc-like structures. In case of Golgi complex, these are stacked parallel to each other like this. Stacked means they are arranged parallelly on one above the other like this. And most important thing, one thing that you need to remember, that this Golgi complex is considered as organelle with polarity. Organelle with polarity. That means the cisternae arranged in such a way that, look at here, can you see this word cis phase and trans phase? That means these cisternae are stacked parallel to each other, and this whole Golgi apparatus exhibits two phases. One is cis phase and trans phase. What is cis phase? Cis phase is also called forming phase, and trans phase is also called maturing phase. That means here, look at here. Now listen to me. For example, it is a ribosome. This ribosome synthesized a protein. Okay? With the help of ribosome, a protein is synthesized. Now this is Golgi apparatus present in the cell. Now this protein synthesized on the ribosomes of endoplasmic reticulum fuses with cis phase and passes through the cisternae and finally comes out as a functional protein or functional component through trans phase. As this protein is fusing with cis phase, It is called forming. As this protein became a functional component and secretes in the form of a vesicle from the transphase, it is called maturing. The cis and trans phases appears to be entirely different, but they are interconnected. Okay, so don't worry about this terminology like cis, trans, and all these things. We are going to explain in detail in our offline classes, because as I'm hurrying in completing, in making you become familiar with many terminology, just I'm delivering, delivering the goods. Only I'm not explaining everything in detail. Just I'm exposing you to different terminology. Now listen to me carefully. So this is about this Golgi complex. Next, when it comes to the lysosomes, lysosomes originates from the Golgi complex, I already told, and lysosomes contain various digesting enzymes, like enzymes that are responsible for digesting lipids, proteins, carbohydrates, nucleic acids. The enzyme which digests lipids is called lipases, proteins, proteases, carbohydrates, carbohydrates like amylase, okay, nucleic acids, nucleases. This all enzymes present. Now this Golgi complex, lysosome enzymes are optimally active at acidic pH. I already told in last before class that these lysosomes are also considered as cell suicidal bags. That means under starvation condition, lysosomal membrane ruptures and its enzymes are released into the cell and it digests the whole cell. As this lysosome killing their own cell under starvation, hence they are called cell suicidal bags. So you may get some doubts like, why in a normal cell lysosomal membrane is not ruptured? Why it is get ruptured under starvation condition, isn't it? So if you know the answer, you tell me the answer at the end of our class, or else you just search it, okay, find the answer and tell me after coming to offline classes. Okay, so this is a small assignment for you. So just think, just go, instead of wasting your time by discussing unnecessary things on the platforms like our educational YouTube channel. So you are even wasting your energy in a place where you are going to procure the energy. See, here you are getting energy in terms of knowledge, right? Even this place are also you are wasting. I'm not saying for all, some people. And remember, this world, this total world is a world of distractions. Remember, be careful. It is a world of distractions. There is no place in this world without distraction. Even in your home, when you come to the offline classes, when you are in online classes, when you are going on the road, everywhere you can find distractions. Then how to overcome this distraction? Overcoming distraction is completely personal. That depends upon your mental status and your stability and your strong determination towards your destiny. For example, if you want to go someplace, say for example, you are coming to a school and your school starts at 8:30 a.m. Now the time is already 8:20. It takes minimum 15 minutes to reach your school, and if you go late to the school, say if your principal is very harsh and she or he don't allow you to into the school, then what will you do? Will you focus on other things? On other things while going to the school? No. Your focus will be only going to the school within the time, like before 8:30. Isn't it? Do you call this preparation, like this focusing on only one particular aspect as a pressure? No, it is not a pressure. It is a preparation for reaching your destiny. Similarly, so now when you are focusing only on particular aspect, all distraction gone out. You are not thinking about this world, and you are not thinking about other aspects. You are not looking anybody, and you are not discussing with anybody. Just you wanted to go to the school, so you are running either by bicycle or moped or with your father, something else, right? So now tell me, who can avoid all your distractions? Is it your surrounding people, or your environment, or yourself? So overcoming from the distractions is all depends on you, not by others. This thing you have to remember. I think you are all clear what I am trying to tell. So don't get distracted for unnecessary things. You are still small kids. Remember, you have, lot of endeavors to face in your life. Just you completed your 10th class. It is a small phase in the entire course of life. It's just one step. Still you have to cross many steps and many hurdles. Okay? So in this process of reaching to the destiny, many distractions will appear in different forms. A boy may get distraction in terms of a girl, or a girl may get a distraction in terms of a boy, or both may get distraction some other way. Like sometimes you feel like to watch mobile, or sometimes you feel like watch a movie. That is also a kind of distraction. Remember, just go through, like even in YouTube also it is available. You just watch some of interviews given by IAS toppers or IPS toppers. Just ask them, or just listen to them, or even you can you can watch interviews of NEET toppers, All India toppers, or IIT toppers. They never surrender themselves to this kind of distractions. Got it? They never surrendered. And they never even touched their mobiles. Still, they reached their destiny. That is the attitude you must have. That's why in very first class itself I told, you must come here with strong dedication, devotion, determination, discipline. Those who have these 4 days can go out with the best medical seat, and they can join with the AIIMS colleges of India. Understood? So I think I myself distracted from this lysosomes topic, because after watching all your messages and chit-chatting among yourself, and I came to know that some teachers are also very harsh on you. It's all happening because of your behavior. If anybody is harsh on you, it is all because of you only, your behavior. It's not, you cannot blame them. So come to the offline classes with 100% concentration only on your ultimate goal. What is that? Getting a medical seat in a AIIMS college, not in local colleges. Okay? So I think I told you that these lysosomes are concerned with digestion process basically. So they also have important role in autolysis process. And apart from that, I also told that these lysosomes in a normal cell, their membrane is not ruptured and enzymes are not released into the cell cytoplasm. Whereas under starvation, these enzymes, released from the lysosomes as their membrane is ruptured. Just I asked you why lysosomal membrane is ruptured and enzymes are released into the cytoplasm when the cell is under starvation, and why this membrane is not ruptured in a normal condition. Okay, if you know the answer, you can tell me now. If you don't know it, then you search it and tell me after coming to the offline classes. Okay, so with this lysosomes, I'll close this session. After this, we'll meet again in our offline classes, most probably after 15th. You may be asked to come for the offline classes. Okay, now your chat box is open. You can ask any doubts that if you have, in our today's class or yesterday's class. Don't ask unrelated, queries. Ask only subjective doubts, like botany related doubts. Okay, now you can ask me. The chat box is opened. I'm waiting for your queries. If you have any query, you can ask me. Otherwise, you can leave the class. Okay, so there is no doubt. Genesis. Bhavikata. Okay. Reporting day June 5th. It was officially announced in our Sri Chaitanya Gosala group. Okay, I don't know. So as per my information that I got today, that I spoke with some administrator today. Just he said that you may be called after 15th of this month. I'm not aware. So you can ask your administrators. Okay, then. Any more subjective doubts? Subject related doubts. And one guy is giving answer to give space for new cells to form. No, that is not the answer. I think it's because of cell cannot support itself, becomes burden on the body or in children. Of course, but that is not the exact answer. You search it and tell me. Okay, you search in books and tell me. Think understanding starvation condition cell releases some enzymes. I'm asking why the membrane is ruptured under starvation. Why is not ruptured under normal condition? And only under starvation alone it is ruptured and it is digesting the cell, not in normal condition. Why? All the topics, which lessons we have to go through 11th NCERT? Yeah, it's a good question. Nikhilesh Ganji. You can just go through, biological classification once you go through. Okay, plant kingdom also you can go through before coming to the offline classes, as we already discussed in our online classes. So just you go through it. Don't read every line, because you cannot understand. There's so much of information is there in every topic and every line. Just peripherally you can read. Okay, next. Why only lysosomes will sacrifice itself to energy to the cell? Not only lysosomes, man. Energy is produced in different ways. Lysosomes, just they play important role in digestion. They are not doing the whole process of digestion. The actual production of energy is carried by mitochondria, not by lysosomes. Okay, Mr. Junior Itachi, Mr. or Miss, I don't know. No online classes will be continued, I think so. If it is continued, you may get another teacher. Might be some mechanism which triggers the breakage of cell wall under starvation. Mr. Karthik, is it cell wall? So no, it's not correct answer. Lysosomes do not rupture in normal because multi-layer protective system keeps digestive enzymes. No, Chalapati Korra, I mean your name is Kiranya. No, wrong, Miss Kiranya. It is not correct, because there might be some mechanism. Your message has been deleted. Sorry for hurting you. Oh, so you hurted me. I did not see your messages ever, never. I don't look into the messages. Only I look only those who are asking doubts related to our subject. So anyhow, your excuse is accepted. Thank you. Thank you for your truthfulness. Not for me, like you hurted many other teachers, it seems. They are also feeling bad. So tell them, sorry for all the teachers, all teachers who came to you in online. May lysosome structure because it becomes, I did not see your, message completely. In which condition cell wall will break? Cell wall do not breaks, it remains. Cell wall breakage is possible with the help of an enzyme, cellulase and pectinase. Okay, that's all. Next. Maybe lysosomes rupture because they become fragile when they are starving. Oh, World Cup 2030. Mr. or Miss, I don't know your name. That is not a correct answer. I need exact answer for my question. That's what you think, and of course you are trying. That itself is a great achievement, a great thing. You are trying to answer my question. You search it and you just go through other books and even our NCERT books. If you find the answer, you can tell me after coming to offline classes. Don't worry. Okay. Sheshacharyalu. See, I did not hurt it at all. So I think you did not cause any kind of disturbance in my class. You are all listening very carefully. So I'm very happy with your behavior. Okay, except very few. But I came to know that many other teachers are, feeling very bad with some of your behavior. So that is not a good thing. That's why I told you that don't deviate yourself, don't distract yourself. Okay, yeah. So with this, I'm closing this session, and all the best for all your future endeavors. And at the same time, heartfully welcoming you all for our offline classes as soon as possible. So I'll meet you all physically in our live classes. Thank you. Bye to all. Goodbye.